Hello, good morning to all of you and welcome to Department of Microbiology, KJ Somaya College of Arts, Commerce and Science, Kopergaon. Let me introduce first myself, Yogesh Chaudhary, and from today we'll be discussing paper MB336, that is Food and Dairy Microbiology. Okay. So, before going to start with actual syllabus, we will discuss some basic things about this paper and syllabus. Okay. So, as you know, that microbes, you know the definition of microbiology, that microbiology is study of microorganisms. Okay. Microbiology includes study of bacteria, fungi, algae, viruses, protozoa. And you know that microbes are ubiquitous. They affect wide aspects of our life. And we can't imagine our life without microbes. Okay. Microbes have both beneficial and harmful activity to us. So microbes are beneficial as well as harmful to us. Okay, so they show both positive and negative activities. Okay, so as you know that microbiology has wide application in different areas. Say for example in agriculture, in environment, in industry. In the same way, microbiology has also played immense role in food and dairy microbiology. Okay, so this role is positive also as well as negative also. Okay, so in this paper we'll see both these aspects. So first let's start with introduction of syllabus. So this paper, that is MB336, food and dairy microbiology, actually it is divided into two sections, that is dairy microbiology and food microbiology. Okay, so in first section we'll see dairy microbiology. In that we'll start with third unit, that is dairy development in India. Okay, so in this particular unit we'll see that in India how that dairy industry get flourished from small scale level up to large scale industrial operation. Okay, so in this particular unit we have to see the role of National Dairy Development Board, National Dairy Research Institute NDRI, then Military Dairy Farm, then Indian Dairy Corporation, then and various dairy cooperatives and milk grades, operations, operation flood, etc. So these are actually institutes of national importance, and these are especially established for development in this dairy sector. Okay. In next fourth unit we'll see some technical details regarding milk, so milk chemistry and its constituents. In that we'll first see definition and composition of milk. So milk, as we know that it is complete food, it has proteins, it has lipids, it has carbohydrates. So we'll define milk and we'll see in detail what are the actual components and what are their functions. Then next we'll see types of milk. You know that milks are of different types, for example skim milk, tone milk, homogenized milk. Okay, and these milks usually in at home we consume raw milk. Okay, so these are flavored milks or some specialized milk types. Okay, then we'll see concept of clean milk. Then next we'll see factors affecting quality and quantity of milk. Okay, and as well as nutritive value, then physicochemical properties of milk. So overall this unit is related to some technical details of milk, like chemistry and its constitution. Then fifth unit, it is related to microbiology. So we'll see in fifth unit how microbes affect, or how what actually role microbes play in milk and in dairy. Okay, so microbes, as I already told you, they play both positive role as well as negative role. Okay, so there are if you consider positive role, then we can know that by using microbes we can produce different types of products from milk. As well as if you consider negative role, so we know that microbes are causative agents of spoilage. Okay, and they spoil the milk. Okay, so in that we'll see first common microorganisms found in milk, as well as fermentation and spoilage of milk. Then various milk-borne diseases. Okay, so this unit is purely dedicated to microbiology. Okay, the next sixth is preservation of milk and pasteurization and its storage. So it is also again very important aspects as far as milk is concerned. Pasteurization, you know, you are studying since your first year course. So in this unit we'll see different method of pasteurizations like LTH, STST, UTH. Then different storage specifications after pasteurization. So storage is also important as well as equal to pasteurization. So we should have some facilities to store milk, specific facilities. Okay. Then there are different tests to diagnose quality of milk. These are phosphatase tests and its significance. Then last unit that in this section, in this first section, is 7th unit, and it is again related to microbiology, and it is microbial analysis of milk. So in this particular module, or this unit, we'll see different types of tests, how we can count microbial cells, or in the how we can conduct different tests to check the quality of milk. In that we first see dye reduction test using methylene blue and research urine. Then total bacterial count. Okay, so this total bacterial count, from this will help that which type and in how much type microbes are present in this milk. Then next is brucellar ring test for mastitis. Mastitis, actually it is disease of mastitis. It is veterinary disease. Okay, it is diseases of cows and buffaloes. Then next we'll see somatic cell count. Okay, so this is overall picture regarding first section, that is dairy microbiology. Now we move towards second section, that is food microbiology. Okay, as you know, food microbiology, you eat food every day. Okay, and food is immense part of our system. Without food we can't get energy. So in this particular section we'll see that how microbes affect this food quality, the storage, okay, and its nutritional properties. So in that first unit we'll see classification of foods based on their stability. So we know that foods are of different types on their stability, like perishable, semi-perishable and stable. So in first unit we'll majorly focus on these types. Okay, then in second unit we'll see food spoilage. Okay, so again this is very important unit with respect to microbiological aspects of food. In this unit we'll see that how microbes affect or spoil the food, how it alters the quality of food. In that we'll see the chemical and physical properties of food affecting microbial growth. Then next we'll see various sources of food spoilage, that how microbes spoil that food, and from which sources that microbes come and enter into the food. In section or part C, we'll see spoilage of meat and various poultry products, then bread, fruits and vegetables, eggs, seafoods, then canned foods. Okay, so overall this second unit is related to microbiology and food. Okay, then next third unit, it is regarding food preservation. Okay, so most of the time, usually what we do, we cook fresh food and we eat it. But in sometimes, in if you go to foreign countries or larger countries, the food, they people use preserved food. Okay, the people use preserved food, and while preserving the food quality should not be altered. It should be safe from microbes. It should not spoilage. So this area has very important as with respect to microbiology. So if you have to maintain quality of food, so you have to take care of food preservation details. So in this, we'll first see the basic principle of food preservation, then various method of food preservation, whether it is cold related or it is temperature related. In first, we'll see thermal destruction of bacteria, that is use of low temperature and high temperature. Okay, then determination of TDP, TDT, D value, F value, Z value, then use of chemicals, antibiotics for food preservation, then canning, it is special type of food preservation technique. Then next, dehydration, that is removal of excess moisture and water. Then use of some radiations. After packing of food, they pack the food, they are exposed to some radiations for killing contaminated microbes. Okay. Then next, principle of hazardous analysis and critical control point. Then next, introduction to Tetra Pak technology. Okay, so overall this third unit is dedicated to food preservation. Then fourth unit, again it is related to microbiology, that how microbiology affects food quality, and it's how microbes poisons the food or how they spoils the food. In that unit we have to see. So food poisoning. Okay, so there are different microbes, they cause food poisoning. For example, Staphylococcus aureus, Campylobacter, Clostridium botulism, Aspergillus flavus. So all these are specific organisms, they cause poisoning of food. Okay, then in B section we will see various food infection. There are sometimes you might heard about food poisoning, food infection. So these are nothing but due to microbes. Microbes spoil that food, and if we eat that spoiled food, then definitely we'll get illness or will get affected. Okay. Then fifth unit, it is dedicated to fermented foods. Fermentation. Okay, so in this fermented food we'll see how microbes carry out fermentation process, what different types of food they produce, or they can alter the raw material into some finished product. So in this we can see definition and types, then significance of fermented foods, that is prebiotic, characteristics of lactic acid bacteria, then fermentation of idli, batter and butter. Okay, so overall this fifth unit is dedicated to fermentation of foods. Then sixth unit, it is some advancement, that is genetic application of genetically modified microorganisms, that is GMOs. So how GMOs can be used in this particular area of food. Okay, so in that we'll start with starter culture, then genetically modified foods, say for example food grade, biopreservatives, then recombinant dairy enzymes, proteins and etc. And in seventh last unit we'll see food standardization and regulation. Okay, so thank you